Okay, so welcome to another Gable Hall practical. This time we're looking at photosynthesis. We're investigating the effect of light on the rate of photosynthesis. We have here a plant, an Elodia plant, or duckweed, which is a pond weed, which grows in water. Now this plant is useful for investigating photosynthesis because then right before our eyes we can see in the form of bubbles when the gas is being released. Going back to our equation for photosynthesis, we know that during photosynthesis water combines with carbon dioxide to produce oxygen, which is the bubbles we're going to be witnessing, and glucose in the presence of light. We're using our lamp here as our source of light, and we're going to measure the distance between the lamp and the pondweed. We have here a table that will help us during the practical to actually record our results. So here we have the distance between the lamp and the plant in centimeters, which is our independent variable, which we change, and the number of bubbles that will be evolved. We're going to trial it first time, second time, third time, and for each of them we calculate the average. This is our dependent variable we're measuring. We're going to keep, obviously, the room temperature within the setting should be the same. We're also going to try and make sure that the plant will remain the same. We're not going to keep changing the plant. Right, so the first time we have the distance measured between our plant and the lamp. We can say this is roughly 10 centimeters, okay? So with our stopwatch, if we agree that we're going to time how many bubbles there are in, say, 2 minutes, we can count the number of bubbles, record that for our trial 1, repeat it trial 2, trial 3, and then increase our distance to, say, 20 centimeters. We are going to do this whole process again. So for 20 meters, we time for 2 minutes, count the number of bubbles, trial 1, number of bubbles, trial 2, trial 3, find the average by adding the 3 and dividing by 3. If there's an outlier, we need to get rid of that and divide by 2 in that case. We're going to repeat this for about 4 more. So suppose we go 30, 40, 50, and 60. That should enable us to plot a graph if we have all our results. Normally, the minimum number of points you should have for a graph is 5. So if we have 6, that should do. Based on this, we can then write a conclusion. If we look at the average and it's giving us an increase, we can say that as we increase the distance, this is what is happening. But really, from our prediction, we should expect to see that the further the lamp is from the pondweed, the fewer the bubbles you'll be getting. Why? Because the light intensity reduces as you increase the distance. Right, I hope this will help with your revision. Thank you.